Hello and welcome to Laboratory 4 in vitro glucosaminoglycan or GAG production by fibroblasts encapsulated in alginate hydrogels. Now that title is kind of a mouthful, so I'm going to use the background section of this lab to kind of break that title down for you into different pieces. First, we'll talk about fibroblasts. The cells that we're going to be using in this lab are NIH3T3 fibroblasts. Fibroblasts are cells found in most connective tissues. They are throughout your body. They synthesize components of the extracellular matrix like glucosaminoglycans, the GAGs, and collagen. But because they're present throughout your body, there's different types of fibroblasts. Fibroblasts from different anatomical locations like cornea, skin, joints, and bones all exhibit different characteristic phenotypes. And one fibroblast line is the NIH3T3 line that was established in 1962 from a mouse embryo by two scientists at NYU. And this cell line has become basically a standard fibroblast cell line that's used in many preliminary studies where you look at biomaterial cell interactions. The reason it's become a standard cell line is that they're very hardy, so they grow easily, they grow quickly, they're relatively economical to purchase and also to maintain. Now let's talk about the glucosaminoglycans that I've mentioned. GAGs are long, unbranched polysaccharide chains that are composed of a repeat unit of a disaccharide. The extracellular matrix contains a mixture of different glucosaminoglycans depending on the tissue location and the age of the person. GAGs have a net negative charge that attracts charged sodium ions, like positive ions. And so therefore, when the positive ions go into the extracellular matrix with the negatively charged GAGs, this causes water to want to diffuse into your tissues via osmosis. So in other words, GAGs help to keep the extracellular matrix and the resident cells hydrated. They also help to bind growth factors in your tissue. Examples of GAGs that you've probably come across in your reading are heparin sulfate, hyaluronic acid, and chondroitin sulfates. Now, scaffolds are central to current tissue engineering strategies. They're usually combined with cells and bioactive molecules to form tissue engineering constructs. Although the field of scaffold-based tissue engineering is still relatively new, there's no clear definition of what the ideal scaffold is. Different tissues and organs have different functional roles in the body, so it's not likely that one single material will serve all those roles. So there's no such thing as a universal scaffold for the regeneration of any type of tissue. However, all scaffolds do have one thing in common. They all have to act as basically an anchor site for transplanted cells, and they also have to produce enough initial mechanical strength to substitute damaged tissues until they're able to regenerate. Now, alginate, this is the same material we've studied in our previous labs. It has a similar structure actually to the GAGs in your body. And it already is used in medicine clinically, for instance, to treat acid reflux, and it's used as wound dressings. And also in the research side of things, alginate is widely studied in tissue engineering, as it has been established to be non-immunogenic. That means it doesn't cause an immune response. And research suggests that it is a viable cell delivery vehicle, or it's viable for cell encapsulation. And it's relatively inexpensive and easy to use, and so that's why we chose to use alginate in this particular experiment. Okay, and just like before, the hydrogels that we will be preparing are made out of calcium alginate. So to get calcium alginate hydrogels, you start with sodium alginate. Again, it's derived from seaweed. When you dissolve it in water, it forms a yellowish, thick, viscous solution, which, when it's exposed to calcium ions, the ions will bond together two chains of the alginate via an ionic bond. And this is called a crosslink, and it causes the yellowish, thick, viscous alginate solution to transition to a soft, solid gel that you can pick up and handle. And in this lab, we're going to be doing something slightly different than before because this time we're encapsulating cells within the alginate, but last time we didn't encapsulate any cells. So what we'll do is we'll suspend the NIH3T3 fibroblasts in solutions of alginate, place them at the bottom of the well, cover them with calcium chloride solution. This will cross-link the outer surface immediately, entrapping the cells within the solid gel and allowing the inside of the gel to become a culture environment for the cells. The cells inside will not only proliferate over time, but begin to synthesize their extracellular matrix. So you'll be responsible for maintaining these cells for 14 days. Remember, you have to feed them and you have to keep them clean. And the end of that period, what you're going to do is stain with alcine blue dye again to detect the glucosaminoglycans that were produced by the encapsulated cells. So in this lab, you're really building on the skills that you developed in labs 2 and 3. And so if you forget what you did in those labs, you can reference those videos at any time when developing your procedures. So for the materials for this lab, each group will receive one confluent flask of NIH3T3 fibroblasts. You'll receive some cell culture medium, DMEM, containing bovine serum and pen strep. It will be warmed for you. You'll need phosphate buffered saline, sterile, alcine blue dye, trypsin, acetic acid solution, ethanol stock solutions between 30 and 100% ethanol, an alginate solution, 1% weight per volume, autoclaved for sterility, calcium chloride solution, 2 weight per volume, autoclaved, 15 ml conical tube, sterile, formalin, 3.7%, 24 well plates, microscope slides, and tweezers or spatula. Now, as far as equipment for this lab, what you'll need is the laminar flow hood, inverted light microscope, an incubator kept at 37 degrees with 5% CO2, a refrigerated centrifuge, and a water bath at 37 degrees Celsius. Now, for the procedure, your goal is that you will be given a monolayer of fibroblasts and you'll want to reseed them in alginate gels for 14 days. You'll start this process by retrieving warm media and trypsin from the water bath. Remember to clean each container before placing in the tissue culture hood. Then you'll take your confluent plate of cells, you'll remove the media, add 1 milliliter of trypsin, and place the plate in the incubator for 5 minutes. Then you'll retrieve the plate from the incubator and add 5 milliliters of warm media over the plate. Remember to gently pipette the media up and down to detach the cells from the plate, and transfer the suspension to a 15 ml centrifuge tube. You can vortex gently to uniformly distribute the cells in the suspension. This is the same exact process we used before when we counted the cells. So after this, you'll use the hemocytometer to determine how many cells are present per milliliter of media. Remember to use the hemocytometer. You just have to remove 10 microliters of cell suspension and add it to each side of the hemocytometer and look at it under the microscope under 10x magnification, and you'll just need to count the number of cells within the defined area, the central square on the microscope slide. And remember, we're adding up the total number of cells in each of the 5 squares highlighted in blue here. This would be equivalent to 1/5 the total volume of the chamber outlined in white, which is 1 times 10 to the minus 4th milliliters. And then you'll find your cell concentration by taking your average cell count from both sides of the hemocytometer, that's N, and dividing it by 1/5 times 1 times 10 to the minus 4th. That'll give you your cell count in cells per milliliter. Now, whatever concentration you get, you'll likely have to dilute your cell suspension. Your target here is to make a cell suspension with a concentration of 7.6 times 10 to the 5th cells per mil. And you'll need 4 milliliters total again. But remember, this is 4 milliliters of alginate solution, not cell culture media, since you want to seed the cells in alginate. So you'll start by doing the calculations just like last time. Use C1V1 equals C2V2. And you're solving for V1 here, which is the volume of cell suspension you need for the dilution. Once you calculate it, remove V1 from the cell suspension and place it in the 15 milliliter conical tube. Now here's where the procedure is a little different than what you did last time. You need to isolate the cell pellet here. So you'll do so by adding a little bit more media, about 5 milliliters, over top of V1 in the 15 milliliter conical tube, and you'll centrifuge at 1500 RPM for 10 minutes at 4 degrees Celsius. This will produce a cell pellet at the bottom of the tube, like the one you saw in Lab 1, and you'll have to do your best to aspirate the cell pellet, isolating the media away from the cells without actually disturbing the cells. And once you isolate your cell pellet, you want to get the appropriate volume of alginate solution into the serological pipette. And then you'll add it over top of your cell pellet. And now you want to uniformly distribute the cells in this alginate solution, so you'll gently pipette up and down several times. And now you're ready to seed that alginate solution into the well plate. And in this experiment, you're going to make approximately 10 samples. And each of those samples consists of 300 microliters of alginate solution pipetted into the bottom of a well. So you'll use a micropipette for that. You'll aspirate up 300 microliters. And with your free hand, you'll open the well plate and drop the alginate solution slowly in the bottom of the well. Remembering that there are cells in the suspension, so you want to do everything as slowly, carefully, and deliberately as possible. Now after you've pipetted all of the alginate solutions into the wells, it's time to add calcium chloride solution over it. And so you'll adjust your micropipette to 700 microliters. And as carefully as you can, slowly drop the calcium chloride over top of the alginate gels. try to pipette towards the corner of the wells to protect the cells. And then when you've finished and you've covered all 10 of your alginate gels, you'll place the lid on the well plate and incubate for 10 minutes. And after 10 minutes, you'll remove the calcium chloride from each well and replace it with 300 microliters of PBS. Then you'll take off that PBS. This is called a PBS wash. And you'll replace with 600 microliters of warm media. Then keep the well plate in the incubator for 14 days. And remember, you have to change the media regularly, at least every Monday, Wednesday, and Friday. And each time you change the media, remember that it has to be warmed to 37 degrees in the water bath before using it, and only open your media bottles in the laminar flow hoods to keep everything sterile. And when you change the media, you'll start by removing 400 microliters of the media that's already there, and then adding 600 microliters of the fresh media on top. Now, after 14 days of culture, you will stain for the GAGs during the regularly scheduled lab period. In order to do so, you'll retrieve your well plates from the incubator. You'll remove media from the plates and add about 700 microliters of PBS, then remove it. Then add PBS again, then remove it. So you'll be doing 2 washes with PBS. After those, you'll add 600 microliters of formalin over the cells and let it sit for about 10 minutes and then do 2 more PBS washes. Then you'll add 600 microliters of Alcine blue stain over the alginate gels. And here's an example of a student adding the 600 microliters of the Alcine blue stain, it's blue as you might expect, over the alginate gel. Now you'll leave this stain over the gels for 30 minutes, incubating at room temperature, so leave the lid on the well plate and leave it in the hood once you're done. And after 30 minutes, you will remove the Alcine blue stain from around the alginate gels. And we really don't want excess stain in the well, so try as best as you can to remove all the excess liquid from around the gel, dumping it into the liquid waste. You may notice that the gels have shrank a little bit, which is normal. Once you've removed all the Alcine blue stain, you'll do your acetic acid wash. So you'll be adding acetic acid to each well. The volume is going to be approximately 600 microliters again. It's enough to cover the gel. And the goal here is to remove excess Alcine blue stain. Now after that, you'll do 2 more washes in DI water, and you'll add 600 microliters of PBS to each well, and wait at least 5 minutes. What this does is the PBS softens the alginate gels, so you'll be able to flatten them easier when you place them on the microscope slide for viewing. Now once your group has secured a spot on a microscope, just use some tweezers or a spatula to transfer the alginate gels to the microscope slide. And here's a picture of an alginate gel that was placed on a microscope slide. You place a second microscope slide on top of that, and you'll push them together as much as possible. The idea here is to get that gel as thin as possible so that light can shine through. And then you can take pictures with the light microscope. Now for the results analysis, present your light microscope histological image of the Alcine blue stained hydrogel. Can you compare these results to what has been published by other investigators? Show relevant data from primary literature and use citations. Can you draw conclusions from your data? What controls could you add to this experiment to strengthen your conclusions? and your response will be graded according to the following rubric. For the discussion, consider this experiment is part of an imaginary project where you are studying alginate as a cell delivery vehicle for any hypothetical application. Give an example of one testable hypothesis that this type of in vitro study could help answer. What controls would you need to add to this experiment in order to prove or disprove your hypothesis? As a tissue engineer, you are interested in enhancing deposition of ECM by hydrogel encapsulated cells. Describe one factor that has been shown to enhance ECM deposition by cells. Formulate a hypothesis and describe an experiment where you would test this factor with cells encapsulated in alginate hydrogels. How will you determine how much ECM was expressed by the cells? And what controls are necessary in this hypothetical experiment? Next, you are investigating alginate gels for cartilage tissue engineering. You just finished the current experiment successfully. That's the experiment we just did. What are the next two experiments that you would design to prove that the scaffolds are viable candidates for this application? What hypothesis does each experiment test? What are the main characterization methods you would need to execute in order to test your hypothesis? Now, describe a study in primary literature where alginate hydrogels are used for cell encapsulation for tissue engineering. What specific application is being considered for the system? Describe the design of the system. How does it work? What are the components? How are the cells encapsulated? And where is it intended to be delivered or implanted? What characterization studies were done? And provide a brief overview of the results. Describe a study in primary literature where alginate hydrogels were made pH sensitive. What application is being considered for the system? Why is pH sensitivity useful for this application? Describe the design of the system. What are the components? Where is it intended to be delivered or implanted? How does it work? What characterization studies were done? And provide a brief overview of the results.